What happens to a world when a creature disappears forever? It is a question that lingers quietly, like the fading ring of a bell long after it has been struck. Does the earth itself remember? Do the forests, the oceans, and the skies hold an imprint of those who once belonged? Or do they dissolve into silence, leaving behind only the stories we tell? As you lie here, letting your breath slow, you imagine walking barefoot across the fabric of time. With each step you come across the shadows of beings that once shaped the rhythm of the planet. Some are familiar, their names carried in the songs of children and the pages of history books. Others are forgotten, slipping into obscurity as though they were never here at all. The first shadow you meet belongs to the dodo. On the island of Mauritius, this bird once moved gently through forests rich with fruit. Its wings, too small for flight, tucked neatly by its sides like folded hands. It had never learned the art of fear, for fear was unnecessary where no predators roamed. But then came ships, sailors, and animals carried across oceans, and the dodo vanished. Extinction arrived swiftly, almost quietly, erasing a life form that had once been abundant. You pause with the image of the dodo in your mind, its round body, its curved beak, its curious eyes. It seems almost to look back at you, as if asking, "Why did you forget me so soon?" The answer is difficult. Extinction feels absolute. It feels like a door closing and locking behind you. And yet, the story does not end there. You continue your walk through memory and come upon the mammoth. The air grows colder around you, as if the breath of ice still lingers in the soil. You hear the muffled thunder of heavy steps, the low rumble of a herd moving across frozen plains. Their tusks curve like crescents of ivory moonlight, sweeping arcs against the snow. For thousands of years, they survived alongside early humans, shaping landscapes as they moved. But the climate shifted, and human hunting sharpened the final edge of their decline. The tundra grew silent. The mammoth disappeared. The thought is almost unbearable, such majesty gone. Yet even in their absence, traces remain. Bones preserved in permafrost, strands of hair still carrying the whisper of their DNA. Inside these fragments lies not only the story of what was lost, but a question of what might return. You let the question hover, light as frost in the air. Could something truly lost ever come back? And if it could, should it? As this thought settles, you lift your gaze to the skies. There, you remember the passenger pigeon. Once, their flocks were so vast that the sun was veiled, and the world below entered an artificial twilight. Their wings sounded like rivers of wind, a living storm that could last for hours as millions passed overhead, and then nothing. Within a century, abundance turned to absence. The skies grew still, emptied of their thunderous wings. The last pigeon, a lone bird named Martha, died in 1914. With her went an entire chapter of Earth's story. You linger in the stillness that follows. The Earth remembers through fossils, through echoes, through the faint imprints left in soil and bone, and now humanity remembers too, through a tool so small yet so powerful that it reaches deep into the spirals of life itself. That tool is CRISPR, clustered, regularly interspaced short palindromic repeats. The words sound technical, almost sharp, but you don't need to hold on to the jargon. Instead, imagine CRISPR as a quill held delicately in the hands of nature itself. With this quill, letters in the book of DNA can be erased, rewritten, or copied. It is not magic, though it feels like it. It is precision, honed by evolution and adapted by human curiosity. CRISPR acts like a pair of tiny scissors. It moves along the twisting helix of DNA, finding the right place to cut. And once a cut is made, the living cell itself rushes to repair, sometimes inserting new words into its code. In this way, scientists are learning how to rewrite the sentences of life, not only to heal, not only to change, but perhaps, one day, to revive. You let your imagination rest on this idea, the possibility of life returning, a feathered shadow taking form again, a mammoth's tusk gleaming once more beneath polar skies, passenger pigeons filling the air in great, sweeping arcs. It is not a certainty. It is not even a promise. It is a possibility. And possibilities are gentle things. They rest on the horizon like the first light of dawn, not demanding, not rushing, but waiting. You feel the weight of questions begin to stir. Would a resurrected mammoth still be a mammoth? Would it carry the same essence as those who walked the icy plains? Or would it be something new, shaped by human hands as much as by ancient DNA? Would bringing back the pigeon change the balance of skies already altered by time? The answers are not here tonight, but the questions themselves are part of the quiet wonder of being alive now, in this moment of possibility. As you breathe, you realize extinction is not just about endings. It is also about memory, about responsibility, and about the gentle longing humans carry for what was lost. CRISPR does not erase extinction, but it cracks open a door we thought was sealed. Through that doorway, imagination steps first, and science follows. Your eyes grow heavy as the thought softens. The creatures remain near you, not fully alive, not entirely gone. They hover at the edge of dream, waiting for the next step in the story. And so you wonder, as your breath slows, if extinction is a closing door, then perhaps DNA is the keyhole, and perhaps CRISPR is the key, turning softly, quietly, almost imperceptibly in the lock. What lies on the other side of that lock leads you deeper into the secret spirals of life's code itself, where every creature, living or gone, carries words written in the same alphabet of 4 letters. You drift with this thought, the dodo, the mammoth. They are also written texts, still encoded, still waiting. And so the journey turns inward, into the double helix, into the very code beneath all life. What if the instructions for making you, every detail from the color of your eyes to the rhythm of your heart, were hidden in a language only a cell could read? That question is not fantasy. It is the reality of DNA. DNA is more than a molecule. It is an archive, a set of blueprints carried inside nearly every living thing. Its form is familiar now. A double helix, 2 strands twisting together like a staircase, spiraling into unseen depths. If you look closer, each step of this staircase is made from paired letters, A with T, C with G, just 4 letters, repeating, rearranging, forming sequences that become the sentences of life. You imagine holding a thread of this code, unwound from a single human cell. It would stretch nearly 2 meters, yet it coils so efficiently that it disappears into a nucleus invisible to the eye. And you are not made of one cell, but trillions, each one carrying the same instructions, the same script, folded and stored with unerring precision. This is the code beneath all life, not just yours, but every bird, tree, and whale. The alphabet never changes, the arrangement does. Like music composed from the same notes, diversity arises from variation. A few switches in sequence can mean the difference between a sunflower and a sequoia, between a hummingbird and an elephant. DNA is not static. It is a record of survival. Across billions of years, mistakes in copying, shifts in sequence, and accidents of chance have created variation. Those variations that allowed a species to thrive were carried forward, generation after generation. In this way, DNA is both present tense and past tense, an inheritance written in chemical ink. Now picture it as a library. Each cell contains shelves of books, volumes that open only when needed. Some volumes describe how to build proteins, the molecular machines that move muscles, carry oxygen, repair damage. Others describe how to regulate when those machines should be made. Most of the library remains closed at any given moment, its words preserved for the right occasion. You are not aware of this reading. It happens continuously, faster than thought. As you breathe, thousands of instructions are followed. As you drift, the code is at work. And all of this unfolds in silence, without fanfare, without pause. Now, imagine being able to walk through this library with a magnifying glass and a pen, to stop at one book, find a single sentence, and change a word. This is where CRISPR enters, not as an abstract tool, but as a means of reading and editing with astonishing accuracy. CRISPR is a system that living bacteria evolved long before humans discovered it. They used it to defend themselves, cutting apart viral DNA that tried to invade. Scientists, watching this natural defense, realized they could adapt it. What began as survival in the microbial world became a way to change the script of life itself. Think of CRISPR as molecular scissors, guided by a search function. It does not cut randomly. It is directed to a specific sequence, like pressing find in a digital document. When it arrives, it makes a cut. Then, as the cell repairs itself, new words can be inserted, a mutation can be removed, a sequence can be rewritten. The scale of this is astonishing. Inside a code of 3 billion letters, the length of the human genome, CRISPR can find a single, misplaced one. It is like standing in a vast library, locating a single spelling error, and correcting it without disturbing the surrounding text. As you let this image unfold, the implications ripple outward. If DNA holds the past, then editing DNA touches the future. What changes are possible when the language of life can be rewritten? Could inherited diseases be removed from families forever? Could crops withstand drought, ensuring food in harsh climates? Could extinct species, preserved in fragments of DNA, step back into existence? These questions do not carry answers here, not tonight, but they rest within you, stirring softly, asking to be considered. You return to the spiral of DNA in your imagination. It feels alive, shifting in slow motion, glimmering with potential. Every rung of the helix is a possibility, every twist a reminder that life is both fragile and resilient. And now, For the first time in history, humanity holds a tool that does more than observe. It edits. You think of it less as a scalpel, more as a pen that can cross out, replace, and rewrite, a pen that hovers over the margins of evolution, not erasing what came before, but altering what may follow. This chapter of science is not about endings, as extinction was. It is about foundations, about the architecture that supports every form of life. It is about realizing that within each of us lies not just a single story, but an entire library waiting to be read. And as you drift deeper into rest, you realize the library is not locked. CRISPR is already holding the key. The spiral closes around you like a staircase fading into the dark. And at its base, you glimpse a new image waiting, a pair of molecular scissors, sharper than steel, yet smaller than dust. How do you fix a story that has a mistake written deep inside its pages? A story not written in ink, but in the twisting letters of DNA that is the puzzle CRISPR-Cas9 was designed to solve. You can picture it as a pair of molecular scissors, so small they slip between the rungs of the double helix. They do not drift aimlessly. They are guided with purpose. They know exactly where to land, exactly which word to cut. The scissors themselves are made of protein, a tool forged by life to protect life. In the world of bacteria, Cas9 evolved as a defender. Viruses would attack, slipping their own genetic code into the bacterial cell, hoping to take it over. But the bacteria kept a memory, storing fragments of those viral codes like photographs in an album. When the same virus returned, Cas9 could be guided by this memory, find the invader's code, and cut it apart, a clean strike, a survival strategy. Humans discovered this system and realized its potential. The bacterial defense became a human instrument, a way to move through genomes with accuracy, to repair or replace what was broken. Imagine walking through a vast orchard. The trees are heavy with vines, twisting up their trunks. Some vines are healthy, strong, and needed. Others are choking branches, limiting the growth of fruit. Cas9 is like the gardener who walks through this orchard with perfect awareness, trimming only the vines that block the light. The guidance comes from RNA, a small strand that acts like a map. This RNA is crafted to match the sequence that needs to be found. When paired with Cas9, it becomes a compass. Together, they move directly to the spot in the DNA where the cut must be made. Once they arrive, Cas9 opens the helix and makes its incision, a precise snip, not a wild slash. Then the cell itself, always repairing and maintaining, begins to heal the break. Sometimes it seals the cut. Sometimes, with guidance, it replaces the missing letters with new ones. The story is changed. This precision is what makes CRISPR remarkable. Other tools came before: zinc, finger, nucleuses, talons, each offering a way to edit DNA. But they were like blunt tools compared to CRISPR's fine blade. Where others were cumbersome, Cas9 is adaptable, directed simply by designing a new RNA guide. It is like changing the address on a letter and sending the scissors to a new destination. Now, pause with this image: a library of billions of letters, a single pair of scissors moving through, searching. And when it finds the exact sequence, it makes its mark. Think of the implications. Inherited diseases are written in the code of DNA. sickle cell anemia, cystic fibrosis, Huntington's disease. Each one is caused by errors, misplaced letters in the script of life. If those letters can be found, if the scissors can reach them, there is a possibility of repair. Not a treatment for symptoms, but a correction of the source. The idea is almost breathtaking, the chance to edit illness out of existence. But CRISPR is not only about medicine. It has touched agriculture, where plants are reshaped to resist drought, to grow with less water, to withstand pests without chemicals. It has reached into laboratories where scientists alter yeast to produce biofuels, or engineer microbes to clean polluted soil. Everywhere DNA exists, the scissors can, in theory, reach. And yet, power always comes with caution. A single misplaced cut could ripple through a genome in unexpected ways. Editing one letter may disturb the harmony of others nearby. Life is not a simple code, but a network, a web of interactions, and so precision demands humility. As you rest inside this thought, you see Cas9 less as a weapon, more as a collaborator. It does not work alone. It works with the cell's own repair machinery, with the rhythms already built into biology. It is like a musician entering an orchestra, not rewriting the score entirely, but adjusting a note so the melody flows. You may wonder how something so small can be controlled. The answer is design. Scientists create the guide RNAs that direct Cas9. By changing only a few bases, they redirect the scissors to a new site in the genome. It is as if you could hand the gardener a list of vines to trim, and each time the gardener would obey. There is awe in this simplicity, a tool so powerful, yet so elegant in its mechanism. One protein, one guide, one cut, and from that, the potential to change the future of species. Still, there are boundaries. Not all regions of DNA are easily accessible. Some are tightly wound, hidden deep within coils. Cas9 cannot always reach them. Other regions may look similar, leading the scissors astray. So research continues, refining, improving, expanding the toolbox. Variants of Cas9 have been created. Smaller versions that slip into cells more easily, softer versions that edit without cutting, versions that simply change a single letter without breaking the strand. You sense the horizon widening. What began as bacterial survival has become a family of tools, each with its own specialty. And this family is still growing. Now, step back and hold the larger picture. You have a double helix of DNA. You have Cas9, the scissors. You have RNA, the guide. Together, they form a trinity of precision. Each plays a role. Each balances the others. The story of CRISPR is not just about science. It is about seeing life's code as editable for the first time. It is about walking into the library of existence, not only as a reader, but as an editor with a pen. You may feel the paradox. Comfort in knowing that diseases might one day be healed. Unease in knowing that the same tool could alter traits, reshape futures, even adjust the essence of species. CRISPR opens a doorway. What lies beyond depends on how it is used. In this space between potential and caution, you rest. Your mind drifts with the rhythm of scissors moving through strands, cut by cut, precise and unhurried. You imagine vines trimmed, orchards opened to light, DNA stories repaired like fragile manuscripts. You feel the possibility, both hopeful and fragile. And as this chapter slows, you sense the next question forming. If the scissors can cut and repair, what else might they create? Can they build, not just fix? Can they write entirely new pages in the library of life? The thought lingers as the orchard fades, as the library grows dim. In the distance, you sense another vision waiting. Not a correction of mistakes, but a redesign of possibilities. A chapter where CRISPR does not only heal, but engineers, reshapes, and invents. Across vast northern plains, silence stretches wide. The air is cold, dry, and still. You imagine a horizon without trees, where grasses once waved like a sea of pale gold. Here, long ago, mammoths walked. They moved slowly, their heavy bodies covered in thick coats of hair, their tusks curving like arcs of ivory against the pale sky. They were creatures of endurance, adapted to the cold, moving in herds across landscapes of snow and grass. To picture them is to picture a time when ice sheets pressed downward, when glaciers shaped valleys, when life survived in harsher climates than most know today. And yet, the mammoth is gone. The last small group, trapped on Wrangel Island off Siberia, faded away only a few thousand years ago. In the span of Earth's history, that is a heartbeat. But in the span of human memory, it is beyond reach. We carry their bones, their frozen remains, their images etched in cave art, but not their living breath. Extinction feels final. When a species vanishes, its songs, movements, and relationships disappear with it. The threads it once wove into ecosystems unravel. The mammoth was more than a giant with tusks. It was part of the grasslands, part of the cycle of life. By trampling snow, they exposed soil, helping grasses grow. By moving in herds, they shaped landscapes. Their absence left quiet changes that spread through the land. Now, a question hovers. Can they return? CRISPR enters this story like a whisper of possibility. Frozen carcasses of mammoths, preserved in permafrost, have kept fragments of their DNA intact. Not whole genomes, but enough pieces to reconstruct. Enough pieces to imagine revival. Here is the process. Scientists compare mammoth DNA to its closest living relative, the Asian elephant. The two are kin, separated by only a few million years. Their genomes overlap closely, but there are differences, especially in traits of cold survival. Thick hair, layers of fat, cold-adapted blood. These are the features written in the mammoth's code. By using CRISPR-Cas9, researchers can edit the elephant's DNA, swapping in mammoth genes piece by piece. The goal is not to copy the mammoth perfectly, but to create a hybrid, a new elephant that carries mammoth-like traits, a creature that could walk through tundra, heavy with fur, shaped for the cold. Not resurrection, exactly. More like reimagining. Pause here in this thought. What does it mean to bring back an echo? Would a mammoth reborn still be a mammoth? Or would it be something new, standing between past and present? Science moves with precision, but meaning lingers in questions. The idea of revival touches something deep in you. You think of stories of lost worlds, of forgotten animals whose bones lie under glass in museums. To bring them back feels like a reversal of time, like turning a page backward in a book that never allows it. It awakens longing for what once was, for what might be again. But revival is not only about nostalgia. it carries ecological vision. Some scientists believe returning mammoths, or mammoth-like elephants, could help restore northern grasslands. By trampling snow, they would prevent permafrost from thawing too quickly. By grazing, they would encourage grasses to spread, which hold carbon in the soil. In this way, the mammoth's return could shape climate resilience, not only a symbol of the past, but an ally for the future. Still, the path is uncertain. The challenges are many. Elephants today are endangered. To use them as surrogates for mammoth-like embryos raises ethical questions. What responsibilities do humans hold to living species, even as they dream of reviving the lost? And if a mammoth steps again onto frozen soil, where will it live? The world it knew has changed. Ice has retreated, climates shift, ecosystems evolve. Can a revived species find a place in landscapes altered by time? These questions press softly, like footprints in snow that slowly fill with frost. They do not erase the vision, but remind you of the weight of it. Picture, for a moment, standing in a snowy plain. The wind moves across open space, and then, in the distance, a shape appears, large, slow, moving with steady rhythm. A woolly figure, tusks sweeping, hair blowing in strands. An animal once thought forever gone, now present before your eyes. The image feels dreamlike, but it rests on real science, unfolding step by step. CRISPR makes this vision imaginable, not inevitable, not simple, but no longer impossible. It transforms extinction from a closed door to a doorway, slightly ajar. Through it, you glimpse futures, both wondrous and uncertain. The science of de-extinction does not stop with mammoths. There are other candidates, passenger pigeons that once filled skies, aurochs that shaped cattle, even frogs that carried their young in their stomachs. Each species tells a story of loss, each raises the possibility of revival, and each presses on the same question: should we? The line between can and should is delicate. CRISPR provides the means, but the choice rests in human hands. To restore a species is to re-enter ecological webs with intentions that ripple outward. It is to intervene in time itself. What begins as science stretches into philosophy, into ethics, into vision. As you reflect, the snowy plain feels less lonely. It holds memories, questions, possibilities. You see science not as cold machinery, but as a continuation of life's own creativity. DNA is a story that does not end. CRISPR is a new author's tool, adding chapters where old ones closed. And in this calm thought, your breathing slows. The mammoth step lingers in your imagination, heavy, deliberate, echoing across the frost. You do not know if it will come again, but you feel its presence, as if time itself remembers. Perhaps this is the gift of CRISPR. Not only the power to cut and repair, but the power to imagine, to ask, to dream of what might walk again. The snow drifts, the horizon softens, and the sound fades. But a question remains, quiet as falling flakes. If we can bring back the lost, what else might we create that has never existed before? The thought glows faintly, like fire under snow. You have walked with giants of the past. Now you wonder about forms not yet born. The next chapter waits, carrying you from echoes of memory to the art of invention. Look upward and imagine the sky filled with motion. Not dozens of birds, not even hundreds, but thousands upon thousands, moving as one. Wings overlapping, shadows sweeping across the ground like shifting clouds. The air alive with sound, coos, wing beats, the rush of bodies passing overhead. This was the sky of North America 2 centuries ago. It belonged to the passenger pigeon. They were among the most numerous birds on Earth. Flocks stretched for miles, sometimes taking hours to pass above a single town. Their numbers were so vast that early settlers wrote of skies that dimmed, as though dusk had arrived in daylight. Trees bent under the weight of nesting pairs. The land beneath them trembled with life. And yet, today, the passenger pigeon is gone. The last one had a name, Martha. She lived in the Cincinnati Zoo and died in 1914. With her passing, a species vanished forever, from billions to zero in less than a century. The loss was swift, final, and shaped by human hands. Hunting was relentless. Passenger pigeons were trapped, netted, shot by the thousands. They were food, sport, and commodity. Forests that once stretched wide were cut down, leaving the pigeons without nesting grounds. Abundance made them vulnerable. What seemed unshakable collapsed quickly. Now the skies feel different, empty, in ways that numbers alone cannot explain. When you look up, you imagine not only what is there, but also what is missing. The absence carries weight. Extinction is often thought of as permanent, but CRISPR unsettles that boundary. With its ability to edit DNA, scientists explore the possibility of bringing back traits of the passenger pigeon. Not resurrection in the purest sense, but recreation. A bird that carries its memory forward into living form. Here is how it might happen. Fragments of passenger pigeon DNA remain, collected from preserved museum specimens. Though damaged and incomplete, these fragments can be compared to the genome of the band-tailed pigeon, the species' closest relative. By lining up the sequences, scientists identify the differences, the unique traits that made the passenger pigeon what it was. Using CRISPR, they aim to insert those traits into the band-tailed pigeon's genome. The result would not be a perfect passenger pigeon, but a hybrid carrying its defining features. If successful, these new birds could one day fly in flocks, reshaping forests in ways their ancestors once did. The passenger pigeon was not just spectacle, it was a keystone species. By breaking branches for nests, it pruned trees. By foraging, it scattered seeds, spreading diversity across forests. Its presence shaped the land as much as the land shaped it. Restoring such a bird would not only honor memory, but also ecology. It would mean giving back a function to forests that still feel its absence. In this way, CRISPR becomes not just a tool of correction, but of restoration. But questions unfold as wings unfold. If we bring them back, where will they live? Forests have changed since their disappearance. Would the new pigeons adapt, or would they struggle? What responsibility would humans hold toward them? To revive a species is to accept stewardship, not only for its survival, but for its role in an altered world. And there are emotional questions, too. What does it mean to see skies filled again, not with the original, but with something close, something reborn? Would the sight heal the wound of loss, or remind us of what cannot truly return? The heart hesitates, even as the mind dreams. Picture the possibility. You walk in a forest in late afternoon. The air feels still. Then, in the distance, a low sound builds. It grows into a storm of wings. Above, the sky darkens, not from clouds, but from countless birds, their movement rippling like a single living fabric. You lift your face, and the past moves. Above you, not as memory, but as presence. That image holds power. It is both a dream and a plan, carried on the edge of science. It suggests that extinction may not always mean silence, that with knowledge, care, and intention, some absences may be answered. But revival is not only about one species. The passenger pigeon becomes a symbol of what is possible. If the sky can be repainted, then the ground, the sea, the rivers may also hold new stories. CRISPR shifts the boundary between loss and potential, between what was and what could be again. Yet, as always, the question lingers. Should we? To restore life is to wield power, and power requires vision, humility, and restraint. Some argue that efforts should focus on protecting species alive today rather than reviving those already gone. Others believe that bringing back what was lost can inspire protection of what remains. Both perspectives circle each other, like flocks moving in wide arcs. You rest within this uncertainty. There is no single answer, only a widening of thought. CRISPR does not erase responsibility, it deepens it. It places the past and the future in your hands, asking not only what can be done, but what ought to be. And so you imagine the skies again, not empty, but alive, not silent, but carrying waves of wings. The sound of billions returning is not yet real, but it hovers as a possibility, a reminder that the world is not fixed, that life can be rewritten, reimagined, renewed. In this moment of reflection, your mind slows. The forest feels near, the sky above you, wide. You hold both loss and hope, as though they are two sides of the same feather. And you know the story of the passenger pigeon is not only about birds, but about humans, about what we take, what we preserve, and what we choose to create again. The air grows still. You hear no wing beats, only the echo of your own breathing, but the thought remains, expanding. If the skies can be refilled, what other forms of life might be painted into the canvas of tomorrow? The sky above you lingers, vast and waiting. You have imagined the return of what was lost. Now another thought takes shape. What of beings that never were, life crafted not from memory, but from imagination? Imagine stepping into a vast garden. It is not an ordinary place of roses, herbs, and trees. This garden grows from possibilities. Every pathway you take opens into rows of questions. Each stem, each blossom, holds a future that might exist or might not. You pause at the entrance, the soil dark beneath your feet. The air is alive with potential, as if the garden itself waits for your thoughts to wander across it. Some of the plants here are familiar, shaped like species, you know. The mammoth, the pigeon, the coral reef, alive again. Others are stranger, almost dreamlike, as though the garden has sprouted forms not yet seen on earth. You move slowly through the rows. On your left, a bed of tall grass-like stems sways. The tags at their base carry names of extinct species: thylacine, aurochs, stellars, sea cow. They are not growing as flesh and bone, but as ideas, held upright by strands of science and hope. DNA fragments, tissue samples, frozen embryos, each One, a seed, waiting for a gardener's hand. CRISPR is the tool that tends this garden. It does not use water or sunlight, but cuts and joins, edits and restores. Through it, genetic information becomes malleable, like clay. And just as a gardener might prune or graft, CRISPR allows the shaping of life at its most fundamental level. Here, the seeds of extinction do not remain silent. They whisper, asking whether they might bloom again. The thought carries both wonder and unease, for in a real garden, not every seed should be planted. Some will not thrive in the climate. Some will grow but spread too far, overtaking what is already there. And some may never be more than symbols, reminders of what could have been. You walk onward, noticing how the garden feels both lush and uncertain. Paths cross one another, leading to sections filled with questions rather than answers. One sign reads, "If we can bring back what is gone, should we?" Another reads, "If we create what has never been, who will care for it?" The garden does not rush you. It holds the questions in stillness, letting them settle into your thoughts like falling leaves. At a bend in the path, you see a tree that is unlike the others. Its trunk is thick, marked with scars that run deep. Each branch carries fruits shaped like possibilities. Some fruits gleam as though filled with light, representing species once lost but perhaps returned. Others are darker, harder to read. New forms of life not yet imagined, still wrapped in mystery. You reach for one of the glowing fruits. It carries the vision of coral reefs restored to color, their DNA strengthened against rising temperatures. Fish dart again among branches of living stone, ecosystems revived from the edge of collapse. The fruit suggests healing, a chance for oceans to breathe more easily. It feels hopeful in your hand. But when you look at a darker fruit, you see uncertainty. What would it mean to design a species that has never existed, shaped not by evolution but by human imagination? A creature built for resilience, perhaps, or for beauty, or even for exploration of places we cannot go. The thought is dazzling and unsettling at once. The fruit is heavy, reminding you that creation always carries weight. This garden does not let you forget the past. In one section, the ground is layered with fossils. The plants here sprout from bones and stones, from shells imprinted in ancient mud. The message is clear. Every future grows from histories written long before. To walk here is to carry both memory and invention. You recall the vast flocks of pigeons, the shadows of mammoths, the forests reshaped by their passing. Yet here, in the garden, they are not only echoes, they are seedlings, waiting for care, waiting for choice. The thought feels like standing at a threshold. One step could open new life, another could leave the soil undisturbed. CRISPR's presence in this place is subtle, like a gardener moving behind the scenes. It does not shout, but its influence is everywhere. Where one leaf is whole, another is carefully mended. Where a stem is broken, new growth emerges, stitched by unseen hands. This is the rhythm of the garden. Precision shaping possibility. You walk deeper, past a section marked imagined futures. Here, the plants look stranger still. One glows faintly in the dusk, as if bioluminescence were borrowed from the deep sea and placed into leaves. Another carries blossoms shaped like spirals, their colors shifting in slow waves. These plants are metaphors for what science may one day allow. organisms that bear no memory of extinction, because they were never part of the past. Life written not as restoration, but as invention. You pause before them, aware of the calmness of the air, and also the weight of the choice implied. What does it mean to become gardeners of futures, not just caretakers of what exists? The question rises like mist and stays with you as you move on. At the center of the garden stands a fountain. Its water flows upward as much as downward, streams crossing and falling back into the pool below. The sound is steady, like the turning of time. Around it, plaques describe different paths: conservation, restoration, invention. Each path has its arguments, its hopes, its risks. And the fountain seems to ask you, which stream will you follow? You realize there may not be one answer. Perhaps all paths matter. To conserve is to protect what we still hold. To restore is to heal what has been broken. To invent is to explore what could exist. The garden offers them all, without forcing your hand. You sit by the fountain, watching the water shimmer. The garden feels less like a place of decision and more like a mirror. It shows you not only what could be grown, but what you are, willing to tend. The weight of choice is real, but so is the beauty of possibility. When you rise again, the pathways stretch before you like sentences waiting to be written. Each step becomes a word, each turn a pause. And as you walk, you feel that the garden itself is writing something through you, a story of futures still unfolding. The air carries no answers, only a sense of vastness, of time stretching forward like soil awaiting seeds. You walk on, carrying images of restored species, imagined beings, and the endless potential of CRISPR as gardener. And in that walk, you feel both awe and hesitation. The garden does not close behind you. It remains a living metaphor, ready to be entered, again in thought or dream. And as you step away, one final question lingers in the air. If life can be tended, rewritten, imagined anew, what stories will we choose to plant next? You leave the garden behind, but the question follows. It hovers like pollen, drifting toward the next thought. For beyond restoring and reviving lies another possibility. Not memory shaped into life, but entirely new sentences written into the code of being. The air feels heavier now. The garden of possibilities is behind you, yet its fragrance clings to your thoughts. As you step into the next clearing, there is no rush of color or motion, only stillness, as though the earth itself holds its breath, waiting for you to ask not what can be grown, but whether it should. This is the place where ethics live, not in arguments written on paper, nor in laws drafted in halls, but in the hush between action and restraint. Here, choices stretch like pathways that do not return once walked. The ground beneath you is smooth, as if worn down by many who stood in this same space, weighing the same questions. You feel the presence of possibility. The woolly mammoth pressing through snow again, the pigeon reshaping skies, the coral reefs shimmering with reborn light. But alongside them rise shadows. What if the mammoth spreads too far? What if the pigeon overwhelms fragile ecosystems that have grown in its absence? What if restoring the reef masks the deeper wound of warming oceans, a bandage over a wound still bleeding? The science of CRISPR is a sculptor's chisel, precise and patient. It can edit genes, cut and paste pieces of the code that weaves life together. It can remove errors, repair damage, insert traits once lost. Yet the question here is not about skill. It is about responsibility. A chisel can carve a masterpiece, or it can fracture what it touches. You picture a long table set in the middle of this space. Around it sit many chairs, each filled by a different voice. Scientists speak of feasibility, of how many base pairs can be restored, how many generations it might take. Conservationists speak of balance, of ecosystems holding both strength and fragility. Philosophers weigh symbols, asking whether bringing back the past distracts us from protecting what still survives. Economists speak of cost, of resources spent on one path that may leave another barren. And somewhere in the shadows, the species themselves, silent, unknowable, wait without speaking. You take a seat at the table, though no one notices. The voices blur, rising and falling like tides. At times they clash, at times they merge. Yet, beneath all their words, you sense the same undertone, the recognition that power has arrived, and power must be carried carefully. Your gaze drifts to the surface of the table. It is covered not in wood grain, but in reflections. You see there the faces of children yet unborn, inheritors of decisions made now. Their eyes do not accuse, nor do they praise. They simply watch, waiting to live with the consequences of choices they had no hand in shaping. The weight of that vision settles into you. Ethics is not a loud thing. It does not rush in with declarations. It moves like a river underground, shaping stone slowly, persistently, until whole landscapes change. And here, in this clearing, you are asked to bend down, to press your ear to the earth, and listen for its flow. You imagine one stream of thought. Bring them back. It flows with hope, with stories of restoration, of mending something we broke. It calls itself justice, a return of what was taken. But another stream flows beside it. Do not disturb what time has already reshaped. That one carries caution, reminding you that extinction is not only loss, but transformation. That ecosystems have moved on, and might suffer if rewound. The two streams never merge. They run in parallel, murmuring against each other, forcing you to choose where to drink. You step away from the table and walk along a corridor of stone markers. Each one carries an inscription: responsibility to the living, respect for the past, humility before the unknown. Together they form a mantra, a guide for decisions that are not only scientific but human. You pause before one marker that glows faintly. It reads, "The line between healing and hubris is narrow." The words sink deep into you. They remind you that ethics is not about stopping wonder, but about asking who bears the burden of its consequences. For example, what if a resurrected species suffers? A mammoth born into a warming world, struggling in climates no longer suited for it. A bird whose flock can never number in the billions again, forever echoing but never whole. Are these lives restored, or are they experiments that cannot fulfill what they once were? The thought stirs unease, but also compassion. You imagine reaching down to lift a fragile sapling from the soil. It may not grow as tall as its ancestors, but your care still matters. Perhaps the question is not whether restoration is perfect, but whether it is kind. Kindness, however, is complicated when ecosystems are involved. Every species touches others. The mammoth reshaped tundra landscapes, knocking down trees, clearing snow to let grasses grow. The pigeon fertilized forests, its droppings feeding soils across a continent. To restore them is not only to revive individuals, it is to stir ancient forces that ripple outward, altering countless lives. And so the question deepens. When we act, are we healing only one species, or reshaping whole worlds without knowing where the changes will end? The corridor opens into a chamber of mirrors. In each mirror, you see a different reflection of humanity. One version is triumphant, celebrated for undoing mistakes of the past. Another is haunted, criticized for arrogance. A third is uncertain, standing forever between pride and regret. The mirrors do not accuse, they simply show possibilities, waiting for time to decide which reflection will come true. You step closer to one mirror, the surface rippling like water. Your own face stares back, but it seems older, marked by years of choices carried. In the lines of your reflection, you see the weight of decisions, never simple, never clean. And in that gaze, you realize something. Ethics is not about certainty. It is about carrying questions without dropping them, even when the answers remain unclear. The mirrors fade, and you find yourself back in the clearing. The long table, the voices, the markers all dissolve into the air. What remains is only the hum of possibility, softer now, more intimate. The clearing does not demand action. It only asks you to remember that power is never free from consequence, and wonder is never separate from responsibility. You stand there, listening, and you realize that ethics, at its heart, is not an obstacle to science, but its companion. It walks beside discovery, holding its arm, reminding it to look not only forward, but also inward. The air shifts, carrying you toward the next thought. If ethics is the companion of science, then what lies beyond? Not only questions of responsibility, but of imagination itself. How we shape entirely new forms of life, not from memory, but from sentences we have never written before. You leave the clearing with its stillness intact, its questions echoing softly in your steps. The path ahead does not return you to the past, but draws you toward a horizon where invention begins. For beyond ethics lies another frontier, not what was, not even what is, but what has never yet existed, waiting for its first breath. You leave the clearing with its stillness intact, its questions echoing softly in your steps. The path ahead does not return you to the past, but draws you toward a horizon where invention begins. For beyond ethics lies another frontier, not what was, not even what is, but what has never yet existed, waiting for its first breath. The path leads you into a vast loom, not of wood or metal, but of roots, rivers, winds, and wings. Everywhere you look, strands cross and knot, forming a tapestry that stretches beyond sight. This is the fabric of ecosystems, woven not by a single hand, but by millions of lives, each pulling, twisting, holding the others in place. You stand at its edge and touch one thread. It hums beneath your finger, alive with energy, trembling with connections unseen. The vibration runs outward, carried along to others, moss clinging to stone, fish gliding through current, seeds riding the air. Each depends on the rest, each sustains something more than itself. You follow one strand, it leads to a frog, perched by a pool in a forest. The frog eats insects, keeping swarms from rising unchecked. Birds eat the frog, their songs filling branches above. The pool itself is fed by rainfall, which depends on trees releasing moisture into the sky. You trace the loop and return to the frog again. Now, not as a single being, but as a node, a knot binding water, insect, bird, and tree. Another thread carries you northward into frozen tundra. Here, you see mammoths of long ago, tusks curved, like crescents of bone, bodies heavy but graceful across the snow. They trample shrubs, knocking down trees, opening spaces for grasses to spread. In their wake, the land shifts, reflecting more sunlight, locking cold deeper into the soil. Their presence is not just life, it is climate, shaping the balance of heat and ice. When they vanished, shrubs spread, forests crept forward, and the tundra warmed more quickly. A single thread, pulled from the weave, left ripples that stretch even to today's atmosphere. You pause, absorbing the scale of it. Extinction is not only absence, it is unraveling. And resurrection, if attempted, is not merely return. It is anew, not tied into a fabric already changed. You turn toward another thread, vibrant with movement. It carries you into skies once covered by passenger pigeons. Their wings beat like rainfall, their flocks so immense that they darkened day into dusk. Their droppings enriched soil, feeding forests across a continent. When they were lost, trees grew differently, their seeds scattered by other creatures, but never with the same abundance. Forests that once leaned on pigeons adjusted, reshaping themselves into patterns not seen before. One thread shifts a forest, one thread warms a climate, one thread changes the rise and fall of insects, the spread of grasses, the arc of rivers. You realize that ecosystems are not fragile because they are weak. They are fragile because they are intricate. Every pull, every cut, every repair alters more than one place. You walk deeper into the loom, and the threads tighten around you, shimmering with light. Some glow like constellations, marking keystone species. The wolves that shape elk herds, the bees that spread pollen, the corals that build homes for countless fish. Others dim and fade, lines once strong but now severed by extinction. And some are newly spun threads of invasive species that entered through accident or design, twisting the weave in ways both surprising and disruptive. You stop before one junction where many strands meet. Here, you see corals beneath the ocean, tiny creatures building vast cities of stone. Fish dart among the pillars, turtles rest in alcoves, currents sweep through corridors, but the coral bleaches, losing its color under heat. A thread weakens, and as it weakens, countless others slacken with it. What seems small, a change in water temperature, echoes across thousands of species, across fishing villages, across the balance of oceans themselves. The loom reminds you of music. Each species is an instrument. Some play loudly, some softly, some only at certain times. Together they form symphonies across landscapes. Lose one voice, and the melody changes. Lose many, and the music falters, leaving silence where once there was song. CRISPR enters your thoughts again, not as a tool alone, but as a hand holding a new thread. Could it reattach what has been broken? Could it stitch lost voices back into the music? The idea stirs wonder, yet the loom answers without words. Threads added now do not enter the same fabric they left. The mammoth returns, but to a tundra reshaped by its absence. The pigeon returns, but to forests that grew in different rhythms. A note can be played again, but the orchestra around it has changed its tempo. You let this awareness settle. Restoration is not a rewind, it is a remix. Ecosystems never stay still long enough for us to return them to what they were. They flow forward, adapting, evolving, reshaping themselves in response to loss and to survival. You sit for a while in the center of the loom, the threads humming around you. You feel both awe and fragility. The recognition that every life is part of something larger, and every choice touches more than it seems. Even small shifts, a pollinator lost, a predator removed, can ripple outward until whole landscapes change. And yet, within this fragility, you also feel resilience. The fabric does not tear easily. It bends, it rewrites, it adjusts. But every adjustment leaves a new pattern, and some patterns carry fewer colors, fewer notes. The thought rises, perhaps the greatest. Responsibility is not only to ask what can return, but how to protect what remains. For each species still alive is already a thread holding others. The bee buzzing in your garden, the plankton in the ocean, the lichens on a stone. Each, though small, is part of a web that keeps the whole world breathing. You imagine yourself holding one thread. It feels light, almost breakable. But as you tug, you feel the pull of hundreds more. It is like holding the whole earth in miniature, realizing that no life stands alone. The loom fades slowly, dissolving into mist. You find yourself standing once more on a path, yet the memory of threads lingers in your fingertips. You walk with the awareness that ecosystems are not scenery but architecture, delicate yet enduring, vulnerable yet powerful. As the mist clears, you sense another horizon ahead. If ecosystems are woven from threads of life, then the next step is not only to restore or protect them, but to imagine what entirely new patterns might emerge. Threads not pulled from the past, but woven into futures we have never seen. You move onward, carrying the memory of threads vibrating in your hands. The weave is fragile, the weave is strong, always shifting, always alive. Ahead lies a space where imagination stretches further still, where life is not only restored or preserved, but written anew as sentences waiting to take form. You rest for a moment, aware of the threads of ecosystems still vibrating in your memory. The weave fades, but in its place, another realization rises. The web does not only stretch outward into the world, it also folds inward into you. Your own body is part of that fabric, carrying not only your life, but countless stories etched into the spiral of your DNA. You breathe, and within each breath, cells stir. Inside those cells, coiled strands of molecules form the instructions for being you. Each strand is a script older than language, older than memory. To read it is to open a time capsule, a record of life stretching back across millions of years. You picture your DNA as a library. Most shelves contain familiar volumes, the ones that keep your heart beating, your blood flowing, your thoughts sparking. But scattered among them are ancient books, dusty with time. Some are still read, guiding your body's work even now. Others are closed, their pages sealed, remnants of stories from ancestors long gone. These remnants are more than curiosities. They are fossils written in code. Fragments of viruses that once infected your ancestors now lie embedded in your genome. Old genes, once vital, sit unused, like tools left behind after the work was done. Each is a trace, a reminder that your body is not only yours, it is also an archive of battles, experiments, and survival across deep time. You walk deeper into this inner library. Shelves rise around you, endless spirals of text. You reach for one fragment, and it speaks of mitochondria. Long ago, these tiny engines inside your cells were not part of you at all. They were free-living bacteria swallowed by ancient cells in a partnership that became permanent. Now they power your every step, but their DNA still whispers of their independence, a fossil of an alliance that shaped all complex life. Another fragment tells of tales. Though you have none now, instructions for a tale still linger within you, a hidden memory of your distant ancestors. In rare cases, those instructions awaken, and children are born with small tales, living echoes of the past. Your body carries these possibilities not because they serve you today, but because time does not erase its notes quickly. You pause at a shelf glowing faintly with something stranger still. Here are viral fragments tucked into your DNA like seeds. They do not attack you. Instead, they lie dormant, passengers in your genetic code. Some even became useful, helping to form the placenta, making pregnancy in mammals possible. Viruses, once enemies, became architects of life itself. The line between invasion and inheritance blurs, and you realize that evolution is not only survival, it is collaboration, theft, and transformation. CRISPR enters this library as a key. It allows scientists to open books long sealed, to rewrite or awaken passages. With it, a dormant gene could be reactivated, a fossil line brought back to voice. You imagine turning pages with such a tool, choosing which ancient scripts to speak again. Would you unlock an ancestral ability? Would you silence a fragment that once caused disease? The choice feels immense, both wondrous and weighty. You walk further, the library shifting into a museum. Glass cases line the halls, each containing pieces of your genetic past. Some cases hold bones, the faint trace of gills that your embryonic body once grew before reshaping into lungs. Others hold feathers of possibility, for the genes that built scales and reptiles also hold the capacity to form hair, fur, or feather. Every exhibit whispers, you are not fixed, you are a mosaic of everything that has come before. In one case, you find echoes of fireflies, not literal but chemical. The same molecules that allow them to glow are cousins to the reactions that power your cells. In another, you glimpse enzymes that once helped bacteria defend themselves against viruses, the very origins of CRISPR itself. Their ancient struggle became humanity's tool. The wars of microbes shaped the possibilities of medicine, and now shape the questions of ethics. You touch the glass and feel time folding inward. Your body is not just the present moment, it is past and potential compressed into the space. Of every cell, you are a bridge, carrying the weight of ancestors and the seeds of futures. The thought steadies you. For if ecosystems are fragile threads outside you, your own body is a web within, woven from histories you did not live, but still carry. The mammoth walks in your DNA. The frog breathes in your code. The pigeon whispers through your spiral. Not literally, but in fragments, in pathways, in echoes of shared origins. You pause by one display, dimmer than the rest. It shows mutations, errors in the script that cause suffering, diseases hidden in the folds of the genome. Some are silent, never awakening. Some reveal themselves in illness passed down generations. Here, too, CRISPR offers possibility to edit, to repair, to relieve. But as you stand in front of the case, the question returns. Not only could we, but should we? To change the text of life is to alter the museum itself. What is preserved? What is rewritten? What is lost? You walk on, the exhibits stretching further. Each reminds you that the boundary between past and present is not a line, but a blur. Your body remembers oceans in its salt. It remembers starlight in the atoms of its bones. It remembers ancestors in fragments of code, still humming, still waiting. A staircase rises before you, spiraling upward like the double helix itself. You ascend slowly, each step carrying you deeper into awareness. At the top, you look back, and the library, the museum, the cases of fossils and fragments, all stretch into the distance like an infinite spiral. It is humbling, and it is beautiful. You realize that to be alive is to be layered. You are the present, but you are also an archive. You are an individual, but you are also a chorus of past lives woven into flesh. And in this recognition, you sense connection, not only to ancestors, but to every living thing. For all creatures carry similar libraries, shaped by similar histories, filled with overlapping echoes. Life is one vast museum, scattered across countless bodies, each holding its own exhibits. You close your eyes and feel it. Your heartbeat is not only your own. It carries rhythms passed through eons, kept alive by genes that have endured through countless storms of change. The present moment feels longer than a second. It stretches backward into prehistory, forward into futures not yet written. When you open your eyes, you notice the library shifting again. The shelves dissolve into pathways of light, leading forward. The exhibits blur into possibilities, as though inviting you to step from history into imagination. For if your body is a time capsule, then what might future bodies become? What stories will they carry? What fragments will they preserve? What edits will they choose to write? The question lingers, slow and steady, not one to be solved tonight, but one to be carried like a thread into the next horizon. You walk onward, feeling your body as both archive and possibility. Each cell a museum, each gene a story, each fragment a whisper of time. Ahead lies a place where this archive becomes blueprint, where the past inside you becomes raw material for futures not yet imagined. The library of your own body fades into the distance, leaving you at the threshold of something new. You have walked among fossils hidden in your cells, listened to the echoes of ancestors, and felt how history lingers inside you. Now, as the path opens ahead, you realize that the story does not end with remembrance. It bends forward into choices not yet made, into futures waiting like seeds beneath the soil. Extinction has long been seen as final. A species disappears, its song vanishes, its form is lost. The dodo, the passenger pigeon, the thylacine, all became shadows in museums, photographs, and fading memories. But science now bends toward the horizon, asking, must extinction be permanent, or can its grip be softened? CRISPR enters here, not as a tool of resurrection alone, but as a tool of protection. It is a way of tending to life before it slips away. Imagine a frog threatened by a fungus that spreads across continents. Within its DNA might lie a dormant variation, a resistance written but unused. CRISPR could awaken that variation, giving the frog a chance to leap again, not into memory but into future forests. Or picture the corals of the sea. Warming oceans bleach their vibrant colors, threatening to erase reefs that cradle countless species. Through CRISPR, scientists explore the possibility of helping corals adapt, giving them resilience to endure higher temperatures, not by replacing the sea's natural rhythm, but by offering a small thread of aid to keep the pattern intact. Each possibility unfurls like a question. Could this tool keep forests alive with bird calls, reefs alive with fish, skies alive with wings? You stand before these questions as though watching dawn spread across the horizon, light touching what was once dark, hope rising with uncertainty. You breathe slowly, and the thought deepens. Extinction is not only about animals and plants. It also shapes human lives. Food systems depend on pollinators, on grains, on cycles of growth that can fray under disease. CRISPR may offer ways to shield crops from blight, to protect harvests from collapse. Such work is not only science but nourishment, a way of sustaining the story of life that supports you with every meal. Still, you feel the weight of responsibility. If life becomes something we edit to preserve, what does it mean to let things be wild? The horizon is not only bright with promise, it is also shadowed by dilemmas. Should we intervene in every struggle? Should we decide which species to help and which to release into history? The questions ripple like waves across your thoughts, neither resolved nor silenced. You pause and imagine a forest. In this forest, some trees stand strong, others falter. A hand reaches in, not to replace the forest, but to steady one sapling, to heal one cracked branch. The forest is still itself, yet it carries the touch of choice. This image stays with you. Science as a gardener, not a ruler, shaping but not owning. From the forest, your thoughts drift to the stars. For as long as life has existed, extinction has been part of its rhythm. Species rise, flourish, vanish, and leave space for others. Mass extinctions reshaped the world, carving paths for new forms of life. If humans now soften extinction's edge, we alter not only the present, but the great story of evolution itself. The question is vast. Are we preserving the past or writing a new kind of future? You sense again the metaphor of threads. In earlier chapters, the web of ecosystems stretched outward. Now, with CRISPR, new threads may be woven. Threads of resilience, threads of survival. The fabric of life becomes not only remembered, but reshaped. The pattern is not finished, it is ongoing, and you hold a place within it. You turn to examples, letting them pass before you like constellations. The chestnut tree, once dominant in North American forests, fell to a blight that spread relentlessly. Now, with CRISPR, researchers work to restore resistance, imagining forests once again filled with chestnut shade. Or the elephants of Africa, threatened by ivory poaching and shrinking habitats. While CRISPR cannot save them from greed, it might help protect them from diseases that add to their burden. Each case is a point of light, small on its own, but together forming a sky full of possibilities. Yet the stars remind you, not all light endures. Even as we preserve, some species may still fade. Science can help, but it cannot hold back every tide. The horizon beyond extinction is not a promise of immortality, but of compassion, of doing what can be done while accepting that some losses may still come. This balance is quiet, almost fragile, and it humbles you. You walk along the path, and the horizon glows wider. You think of the children not yet born, who may live in a world where passenger pigeons fly again, or where forests bloom with trees once thought lost. You think of them breathing air filled with songs and scents preserved by science. You think also of their questions. What did it mean to change life so directly, to reach into the spiral of DNA and leave a mark? They may thank you, or they may wonder at the weight of those choices. Your footsteps slow, and you notice the horizon has a rhythm. It pulses like a heartbeat, steady and alive. The future is not something waiting far away. It is beating here, in the present, in the choices unfolding now. Science is not separate from life. It is a part of life's own striving to continue, to endure, to adapt. And you, listening in the stillness of this night, are part of that pulse as well. You pause once more and lift your gaze. The horizon glows with color, not only of sunrise, but of possibility. Extinction once meant an ending without return. Now it holds the shimmer of continuation, of stories prolonged, of lives extended. Not every story, not forever, but more than before. You breathe this in, slow and deep. You feel it settle in your chest, the mingling of hope and caution, of wonder and humility. The thought is steadying. The horizon is wide, but your place within it is small. And that smallness is part of the beauty. You are one listener, one dreamer, one caretaker among billions. And yet, within that, you hold the power of reflection, of care, of choosing how to step into tomorrow. As you walk, the horizon shifts again. It is no longer only light. It is also vast space. Beyond extinction lies not only survival, but imagination. What might life become if given the chance? What forms might emerge? What songs might rise? What patterns might unfold if the threads are not cut too soon? This final thought rests with you. Science can preserve, but it cannot predict all that will bloom. It can open doors, but it cannot walk through them alone. Life itself must step forward, carrying the edits, the protections, the gifts into futures unseen. And so you watch the horizon, not as an answer, but as a question, wide, luminous, and waiting. You let the horizon hold you. Extinction softens into possibility. Endings blur into beginnings, and the future unfolds like a sky filled with dawn. You rest in that glow, feeling both the weight and the wonder of life's fragile thread. And as your breathing slows, you drift into sleep, carrying the horizon with you. Not as a conclusion, but as a quiet invitation to dream of what might come next.